Okay, so this is all new to me. So Alberto has been heavily involved with this since its inception. So I will be coming up to speed on all the things that are going on. So the nice thing about that is you're actually going to get a very fresh perspective because I don't actually have any other perspective. So, so for a little bit, for an update, I think the efforts that have started here are really great. I think it's important. You know, we in the diagnostic field, especially in the in vitro diagnostic field, believe that we are at the forefront and the beginning of innovation. We're at the forefront of healthcare, and having this partnership will help us all do our jobs better. So I think one of the nice things about this effort is it really is about harmonization and actually making things more transparent to people that will actually help everybody move forward. And it's going to get everybody moving forward on using the same playbook, which I think is really important. In the government, or at least in the work that we do, we kind of work really well one-on-one with most companies. So because you know, everybody's a little bit different. So we meet individually, we give good advice, maybe sometimes not so good advice, but we always give advice, and it's all individual. And that group goes away, and then we're giving the same advice again, or a little bit different to somebody else. So we do that over and over again, so it's not terribly efficient. It tends to be very helpful and productive for the people that we're interacting with, but there's so many people out there that can actually benefit from all of that. So I think having what you're going to hear about today from April is just the beginning of things that are actually making things more transparent and more useful for everybody to use. So the clinical diagnostic program has actually, I think they've got 4 different projects going on now, and April's the winner. Hers finished first. So although as a group effort, it's always good to have strong leaders also. So you'll be hearing more about that, so I don't really want to talk about that. But there are other things going on, too, and it's kind of like one is called clinical evidence, and so I look at that and it's like, okay, that's a pretty big topic. I'm not quite sure how you're going to tackle all of that. But I think there's lots of things in our world that actually would be helpful. So hopefully I will be able to help steer some of this to things that I think will be helpful for everybody, because part of clinical evidence, most of in vitro diagnostics and things in the diagnostic world are actually class 2 products. So it's really about comparisons, so comparative studies and stuff like that. So I think for us, from my perspective, actually finding better ways to ensure consistent reporting of comparison types of studies and stuff, I think would be a nice way to complement this effort. And because everybody does that, then pretty much the whole, if you're not exempt, you're probably a 510K unless you're a companion diagnostic. So then in capillary blood collection, finger stick, it's interesting because you guys think of this, you're approaching this a little bit different from how FDA would approach it. So within our office, we've been struggling with, point of care testing, you know, that's the future. You're getting things closer to the patients, closer to the bedside. So all of that is really important to us, and we're kind of struggling with, okay, well, how do we ensure that moves quicker? How do that moves better with wave testing and all these other things? How do we do that? So we look at it as this huge area of point-of-care testing. I'm not exactly sure what your capillary blood finger stick testing is, but it's actually a nice way to break that whole point-of-care testing into pieces, and actually pieces that you can probably chew, and we can probably develop good information for companies, useful information for FDA to provide feedback back, and as well as useful information for FDA reviewers, which of course, with that in hand, you're actually looking at benefiting patients. So I see it's an interesting take on something that we constantly think about. And then IVD real-world evidence. I'm going to keep my fingers crossed for you all, because I'm not quite sure how we'll do that, but I hope we do. So I kind of like Jeff's statement. It's like, no, let's get rid of all these terms and whatever. It's actually about doing innovative things to get things out in the market faster. So you want to call that real-world evidence. You want to call it, you know, whatever you want to call it doesn't really matter to me. It doesn't actually matter to FDA. It's actually about doing good science and actually having providing benefits to patients. And I think that we're all here for that. So, you know, I like this effort because MDIC has already had some successes. Patient preference, I think, being a big one. So I hope that this group will actually build upon those successes. And I think starting off with the effort that April's going to talk about is a really good start. I was talking to Carolyn earlier. I was like, I'd like to see that to be the start because I actually see, you know, surrogate samples are really important, and that's really nice, but I'd actually like to see us take it, you know, a little bit further. So that's, you know, there are lots of things that could complement that. So you know, in IBD's world, it's like bank samples, archive samples. That would be a nice complement to that effort. Talking about precision medicine and everything, in silico samples, that would be fabulous to get efforts going on in that area for everybody. So we probably don't have as much experience there within the agency, so it would be really exciting for us to work with everybody in that area too. So hopefully I see the efforts that have begun as a foundation and as building blocks for the things to come. So with that, I will kind of stop. I've kind of reached my level of creativity, and I'll introduce April.
Thanks, Don, and thank you all for attending today to hear about the Surrogate Sample Framework. We released the framework last week and are really pleased to have this opportunity to talk about it. I myself am pleased to speak on behalf of our Surrogate Sample Work Group. So why this initiative? What led us to this point? In terms of IVD development and validation studies, there's a heavy reliance on biological patient samples in order to conduct those studies and do that development. When these materials are in short supply, there is a role for surrogate materials or surrogate samples to help accelerate the timelines of development as well as the efficiency of the IVD validations, ultimately benefiting patients with more timely access to in vitro diagnostics. The work on this project was conducted by a talented group of individuals from government, industry, and we had our subject matter experts as well. And I wanted to just take this opportunity to acknowledge and recognize the contributions and dedication of the team in terms of formulating the framework and meeting our timelines to deliver it to you. So why do firms or individuals or developers turn to surrogate samples? Surrogate samples fill a gap. So when an analyte is a rare analyte and difficult to obtain or acquire, there's a role for surrogate samples. At other times, there may be a need to cover the range of an assay. So there may be a need to supplement samples in the high or low range of an assay in order to conduct studies or the medical decision level point. Certain studies require a certain volume or quantity of samples in which to conduct the studies. And that cannot necessarily be satisfied by a patient, a single patient sample. In other cases, there are ethical challenges that lead one to move towards the use of surrogate samples. For example, there are ethical considerations related to taking large volumes of sample or repeated sampling of sick patients. So for these reasons, developers will frequently turn to the use of surrogate samples for parts of the IVD validation studies. So before I delve into the details of the framework, I wanted to just spend a few minutes talking about what it is and what it isn't. First and foremost, one of the items that the work group focused on was setting forth common language and terminology. So we have a definition of surrogate sample and associated terminology. The framework strives to create a balance between general approaches which can be applied across the diverse range of diagnostics, as well as study-specific approaches. There are certain areas where there just was not a general approach, or when we considered certain study types, there were some specific items that needed to be addressed differently. The framework is not focused on study design. So to the extent that study design is important in understanding a particular study type and the surrogate sample usage, it does talk about that. But there are existing guidelines, standards, and FDA guidance document on study design, and we recognize that. Lastly, the framework does put forth a hierarchy, a logical way of looking at the use of surrogate samples and going through a stepwise process in terms of determining what is most appropriate in a particular situation. The elements of the framework consist of a uniform definition of surrogate sample, which has been supplemented with a diverse array of examples to help illustrate the concepts and the points. The framework outlines general principles and considerations when designing and selecting surrogate samples, and it also presents a general hierarchy to aid in the decision-making process. Lastly, it establishes study-specific principles. So there are study-specific principles which have been accompanied by a hierarchy for each study type. So when our work group first came together, one of the challenges that we actually had was conversation and getting clarity as to what it is we were actually talking about. There's a lot of different terms that are used in the industry, contrived samples, altered samples, things are supplemented and pooled. So we started off on looking at getting a common definition, a common term for use in this area. We did look to existing standards and guidelines to source existing terminology where we could, but there is no definition, or there was no definition of surrogate sample, and therefore the team put forth the following definition, that it's material or combination of materials that is used as a substitute for body fluid or tissue taken for examination from a single human subject to study the characteristics of interest. We felt it was also important to supplement the definition with some examples. These are not all inclusive. They might include materials that are supplemented or spiked with an analyte, or materials that are pooled together. Artificial material that's created to represent the functional characteristics of a patient specimen would be another example. There were additional terms which were used quite frequently in this space. Supplemented, which is spiking a matrix with an analyte of interest. Pooled, which is combining 2 or more patient samples together, and that may include spiking or supplementing as well. And then stimulated, which we've defined as including altered materials or materials artificially created. So altered materials of biological origin would include items such as biological material that's been chemically treated or heat treated, and then the artificial materials are those that are created to represent properties of the human specimen. The framework establishes a table of surrogate sample combinations. The table consists of, at the top, matrix. So matrix might be biological or it might be artificial. Within the biological matrix, it might be unaltered or altered, again, such as a heat-treated biological matrix. The matrix may be an individual specimen or it may be pooled combinations of 2 or more human specimens. And then on the other parameter that is considered is analyte. So analyte may be added to the sample or it may not, so that's where we have unspiked. And then when analyte is added, supplemented or spiked, it may be of biological origin or it may be artificial. We added alphanumerical numbers or identifiers to aid in the discussion. They don't have any inherent meaning, but they are there to help facilitate the use of the foundation as one goes through the document with reference being made by these identifiers. The framework, as I said, moves into general principles, which would be applicable across the wide array of IVDs. And the framework principles begin at a high level at the top, identifying the objective for using the surrogate samples is one looking to supplement or actually replace human specimens. What are critical factors related to the performance study type? For certain performance study types, one is using surrogates perhaps to meet the demands, the scientific demands of the study, such as sufficient volume or quantity, whereas other study types, it's key to maintain biological variability of individual patient specimens in order to properly conduct the study. There may be unique factors to consider related to whether the test is quantitative or qualitative. And then there are other factors that need to be considered when there is a sample processing method intended for the specimen, incorporating that and addressing that in the preparation and design of the surrogate. In terms of selection of a specimen matrix, when one is mimicking a specimen matrix, consider what are the most relevant characteristics. For example, viscosity may be important for certain specimen types. Consider those factors when the surrogate sample is being developed. There are different analyte types, and these may confer bias, either positive or negative. So in going through the process, this is an additional point which needs to be considered. And then lastly, assess the combination of matrix and analyte. In certain instances, depending on the test type, like an immunoassay versus a molecular-based technology, there may be more of a reason to ensure that the matrix is biological or the artificial item versus the analyte. So these are pieces that would be considered as well. Again, recommend existing guidelines be consulted where they do exist, and looking at a hierarchical approach to the selection. The surrogate sample hierarchy, so this is the general hierarchy. It builds off the combinations table. In the dark green, we have patient sample. So again, we're starting with a top-level approach. If one were to begin with a patient sample and then that is not available, looking at the various studies, we have found that one either branches off into supplementing individual patient samples or moving towards pooled samples. And so the two arrows here denote where one would begin in the assessment process. In terms of the other items, we found it challenging to come up with general recommendations which would apply across the different study types. And therefore, in terms of assessing and making choices, we took on the study-specific hierarchy as a means to provide additional guidance in this area. There are two other areas on here that are shaded, and that relates to the two types of analytes. We also wanted to convey with the hierarchy that there are certain principles related to preparation, which can influence the ultimate outcome. So part two of the hierarchy focuses on an order of preference in those cases where one would be supplementing a sample or preparing dilutions. And this is in order to preserve matrix integrity and to minimize the effects of dilutions. The hierarchy addresses 9 performance study types, which are listed here on this slide. Each of these sections consists of a narrative describing the principles and how they relate to the particular study type. And then they are also accompanied by the next level of detail, which is the study-specific hierarchy. So this is the study-specific hierarchy. For each of the study types listed, a column was created, and then the identifiers here relate back to the combinations table and have been presented here for conciseness in that manner. The dark green represents patient sample again, and then the hierarchy moves in a linear fashion for consideration. So the medium shaded green area represents those areas where one would consider the selection of a surrogate using a approach that moves downward in a linear fashion. For those areas where there's a lighter shaded green text, these are areas where it was difficult to just say for all studies we're going to move from this point to that point. And depending on variabilities related with the study or the analyte type, the assay type, again, immunoassay versus molecular, there may be different reasons to go with one choice over another. So we wanted to designate that in with the shading. In terms of the peach-covered area, these are areas where we recommend considering other options before approaching these types of materials as a surrogate sample for that particular study type. And then the columns that end earlier, which are denoted with an asterisk, represent those areas where, based on our analysis, the remaining surrogate sample types may not be the most appropriate for that particular study type. So this was intended to convey that information so as not to steer people in the wrong direction. It's recognized that there probably is an exception and that those exceptions may very well be valid, but as a general rule, going down a particular path with that study type would not be the approach we recommend with the foundation in this framework. So as I started off with, the framework is final and it is available. It can be accessed via the MDIC website. I'm very pleased on behalf of the Surrogate Sample Working Group to have this opportunity to present it to you. And I will now be turning it over to our moderator for the panel. Now.
Can I first ask you a question?
Sure. Go ahead.
So what is the next step with respect to the surrogate sample framework?
The next step is to look at the creation of training material. So our work group will be reconvening. Our goal is to create training material reflective of the foundation that would then be used by multiple stakeholders, such as by FDA, by industry, that we're all starting off at least from a base point in terms of the training materials.
And then the next step after training, I'd like to get it to a standards organization and have it be turned into a standard.
Well, see, that's what I was hoping she was going to say.
I'm always way ahead of them.
I know. You were already laying out your wish list before I could even ask the question. I hope somebody's taking really good notes because we're going to be getting Don's wish list throughout the day. We have a great group of panelists. Their names are up here. Vicki, Anastasi, Steve Binion, Jaime Houghton, Don St. Pierre, April Veoukas, and Jeff Zinza. So they'll be joining in this discussion today. So as Don already went through, there are 4 key areas in clinical diagnostics that MDIC is focused on. You've just heard about surrogate samples. We've talked a little bit about the application of real-world data and real-world evidence in the IVD sector, which is the one that I'm leading. So, Don, it has to go well. And then the finger stick, which Don already mentioned, as well as clinical evidence. So I want to ask the panel, how will your organization benefit from the current programs, clinical diagnostic programs that MDIC is doing? Let's start with Steve Binion.
All right. Thanks, Danelle. So the 30-second infomercial, BD is one of the founding members of MDIC from the industry side. Honestly, we're looking for similarly impactful, if you will, outcomes from each of the work groups. I think we've got folks participating in certainly the RWE as well as what Don has sort of called the shorthand for the point of care diagnostics, and just consistent with the output from the surrogate samples team. We're certainly looking for technical tools, guidance, but not in the regulatory definition that we can employ. So.
Great. Vicki, can you just continue?
Sure. So first on, I have clinical evidence. So I'm cheering that one. So we're looking forward to getting that in front of Marina very soon, and then moving that forward. So it's been fun. Talk about definitions. Where's April? That was a fun one. So I'm from ICON, and we're a CRO. So we work with most people in the room, or we interact with most people in the room. So we see everyone's quandaries across the board. So what we're hoping to get out of these is a platform, a framework that will add consistency, and that we can start seeing the industry come together and create programs that will allow the product to go forward faster.
Terrific. Seth, how is Hologic looking at it?
Well, we recently joined the initiatives over the summer, or just last summer. We participated in surrogate samples at a late stage, mainly in a reviewing capacity, just to make sure that our needs weren't perhaps overlooked, but there was really good participation from molecular diagnostics on that working group that helped make sure that our interests were so that was very helpful for us. But that was sort of a baby step for us to get involved on the clinical diagnostic side. We definitely are using those concepts in our current interactions with FDA. Having a framework where industry and FDA have participated together in its development, I think is very helpful for us. It gets that buy-in, perhaps when there might be skepticism on certain approaches. I think that's going to be really helpful for us. In the future, definitely we're going to be involved in IVD clinical evidence and RWE. Those topics are very important for us, and we wouldn't want to be left behind while the industry moves forward on that. So participating, I think, is very important at this stage. Another area I think that was important to us, but I'm not sure will continue, is incremental improvements. That's something that is very important to us as a company that has a lot of legacy products that are utilized in cytology and molecular diagnostics, and often those workflows connect. And when we're looking at improvements made on one side, we've got to make sure that the improvements correspond on the other side, revalidating each set of products each time. Very nice.
Great, thanks. Jaime, what about Sysmex?
Jaime Houghton from Sysmex, and yes, we've been part of the
working groups as well, participating in several of them. I think just as everyone else has said, really the framework for us to have discussions as we go in, looking at the studies that we need to do, understanding what samples we need to have, different approaches for that. I think we're still looking through the real-world evidence to understand how we might be able to leverage that. But certainly, I think that's, continuing to participate and be active in that.
Yeah, and Sysmex is active in that group as well, and we're looking forward to Hologic joining that group.
Thanks.
April, what about Abbott? From our perspective, we're very happy to be a part of the different initiatives. We see, for example, the Surrogate sample framework as a means to help facilitate more efficient conversations with the agency. It's actually a tool that can help with those conversations, assure we're aligned and when we're discussing something, that we are all discussing the same point. That we're not, walking away with different interpretations of the same of a concept. Real-world evidence, again, this is an area of opportunity. So really looking at how this might be applied to diagnostics. There is, quite a bit of effort in other areas of the device space, but having an example or a case study, a use study on how it would apply in the diagnostic area, we believe would help further the advancement in this area. In terms of the finger stick proposal, we are, again, being in rapid diagnostics, it has a direct link to the ability to move those types of products to patients at a more rapid pace when the key details in terms of design are identified and documented out in a tool, as opposed to having to approach each item on individual, with individual product areas. So all in all, we see some specific benefits in all of these areas, and look forward to the completion of projects in the other work groups.
Great, thanks. And Don, I know you're very new to this new position, but do you have any thoughts on how FDA can really benefit from these projects? I know you've mentioned a few, but.
So I'd say probably already benefiting, because if I take up on Steve's comment, which I have to say, whoever set these cards up here did a really nice job. Steve and I go way back, so it's nice having us at different ends of the table. So one of the things that Steve said was, if the framework is nice to have out there as kind of a guideline, but not as a guidance. Well, if it was a guidance, it actually wouldn't be out. We'd still be working on it for a few years before it got out. So that, to me, is a real benefit, being able to actually get things out that actually help people. And, some of the other comments you talk about, kind of like these incremental improvements and stuff like that. I can retire next year, so we're done with incremental improvements. Let's kind of move. So let's see about how we can make things not incremental and make those differences. So I think that's a huge challenge, and so I'm looking forward to that. April's smiling at me like, oh, boy. I don't want to be on that next work group.
Yeah. Well,
so,
I think she was hearing that I can retire in a minute.
So I think there's great opportunities, and it's I've kind of always said historically, it's I absolutely love the FDA, and I think FDA does really good work. I think we do really much better work when we collaborate with industry and with patients and with other people. So I think this is a real opportunity to do that, and it's a bigger opportunity than we've actually had in the past. So I'm very excited about it, and I'm very happy that Maureen is going to be doing a lot of the work.
Okay, I'm going to keep you talking, Don. Does anybody have anything to add to this? No? Okay. Well, I want to keep you talking.
Thanks, Don, for those kind words.
He really meant that he likes you so much that, Okay, so Don, you already mentioned perhaps some areas in which MDIC could go. But what I'd really like to know is what keeps you up at night in terms of these types of issues? How do we get technology out, et cetera? So what's keeping you up at night, besides your new job?
So Alberta's retirement is keeping me up at night, yes. My year-and-a-half-old is keeping me up at night. Other than that, nothing's really keeping me up at night. I have to say, it's been great working in the medical device area, in the healthcare area. People, this is an industry that can, certainly mostly regulate itself. It's an industry that cares about, patients. It makes a difference in the world. And it makes a difference in the country. So I don't actually stay up at night at all, other than thinking I'm really happy about being part of it. I have the thing that keeps me up at night is actually not keeping people excited about government and about government work, because I think the government can actually make lots of things happen. So I think we're going to try to help make lots of things happen.
Good. Good to hear. April, in terms of bringing your technologies to patients, what's keeping you up?
I think the areas that we see opportunity and benefit are in having the common frameworks and tools to make the whole process more efficient. Not having to repeat a study, for example, is big benefit, and having ways to address items in that more efficient manner. In the area of maybe new markers, looking at opportunities to get to the right study design and the right level of evidence in order to bring those products to patients at a more efficient pace would be another area as well.
Jaime?
So I'd say for us, one of the biggest challenges, knowing that our technology is pretty similar, honestly. Sysmex right now is very hematology focused for many, many years, and the technology is about the same. So as we bring new devices, right, we're looking at the studies that we need to do, and really it's availability of samples that becomes a very difficult issue for us when they're required to be fresh samples. And it's not like as far as the sites you can go to, you need to be at a site that has access to an array of samples, so from the high end to the low end, and those don't exist at just any old lab. So as labs continue to do more with less people, to find that lab that has the availability of the samples and the time that they can commit to actually doing the studies, I think for us that continues to be a struggle.
Okay. Jeff?
So when we develop innovative products, we have to steward them appropriately, and there's lots of potential uses for our technologies, but not all of them are validated. And when we see that validation requirements may be more than we can justify economically, we see labs take matters into their own hands. And because of existing policy in that space, we can't offer them the support that they would need as the we can't give them DHF documentation that would really help them to do it appropriately. So I guess what keeps me up at night is knowing that we could be doing more, but we don't because of the difficulties in collecting the data to validate those new uses.
Good point. Vicki?
So I'm not with the company again, I'm with the CRO. So
we
see all these struggles out there. I think I would echo what Jaime said. When we have many studies going on at the same site, looking for the same samples, looking for the same short time period to utilize the sample, and the ability to think beyond that, maybe use global sample collection, look at minimizing samples based on population. I think those are the types of things, and if you look over on the device side, you've seen a minimalistic approach to study design, whereas what's the least burdensome amount of evidence that we need. And I think that in the diagnostic space, as we have so many companies competing for the same sample, literally for the same patient, for the same sample, for the same sequence of samples from that one patient, I think we have to be realistic about how much data we can get and how much is really needed in order to get these products out there in the market and maintain the competitive landscape. It's important for where we are.
Oh, thanks. Steve, what's keeping you up at night?
Can't be me, Steve.
Oh, yeah. Thinking actually less in terms of, BD as a company, more about the MDIC initiatives. I think in the clinical diagnostics area, something, I don't worry about it, I just hope it'll continue to play out, is ongoing, thank you, Marina and Giovanna and others, and even increased commitment within OIR to participating. I think that's been a critical aspect of the successes to date. Another topic area that is, to my knowledge, unresolved. Carolyn and others were instrumental in teeing up some discussions, largely, under the MDIC umbrella, regarding interoperability in IVDs. And I think, going forward, if the RWE initiative is really going to come to fruition, that's going to be a important part of that. One other, and then I'll shut up, is I think we do need to, it's hard to strike the right balance between the technical tools versus guidance. But I would love to see MDIC in the clinical diagnostic space continue to focus on tools that could or would potentially impact and create a more level playing field between LDTs and FDA-regulated IVDs. So,
great.
Other than that, I sleep like a baby.
Well, one thing that you guys didn't throw out that I'm going to throw out is digital health in the IVD space. Is that, it's moving so quickly in terms of innovation and technology that I think it's challenging all of us to work together to find out, how to regulate it, validate it, et cetera. I mean, when I think of artificial intelligence and use of artificial intelligence in clinical decision support or in software of other types, that sort of thing is a challenge for all of us. And so I think, to the extent that something keeps me up at night, that's probably in that space. So now let's talk a little bit about solutions. Okay, we've talked a little bit about what's keeping us up. Well, what regulatory science tools do we think would resolve that? So, Steve?
Mention interoperability again.
Yeah.
I love that.
Well, he's going to say he wants LOINC code.
I can't even spell that.
LOINC.
Wrong. Now, actually, in all seriousness, when we had the prep for this panel, Don agreed that a la C-SPAN, I was going to yield my 5 minutes to him. So,
nice try.
I think in terms of specific tools, I don't have any, new toys, so to speak, to bring to the table. Certainly, the surrogate sample, framework is a good example of an application. And perhaps it's not quite at the level of specificity of a tool, but I do think that MDIC could continue to serve as a forum for discussion of ideas. And I think it's probably, I think just as a matter of process, periodically or even routinely, we should go back to the early brainstorming, stakeholder discussions that actually, I think, led to the current projects within the diagnostics sector, if you will, and take a fresh look.
Yeah, and I think that's a good way to frame it, I think, is it doesn't need to be a tool, but what is it that you think we need to look to, whether it's a topic, whether it's a process, et cetera.
Well, the ones I mentioned, and I think your addition of digital health is probably enough.
Great. Vicki, what do you think?
I think we need to find a way to use electronic health records in diagnostics.
Yeah.
I think, if the rest of the world is using that and we're finding real-world evidence, we're making great strides, and there's a big gap right now with diagnostics, for the ability to find mechanisms and tools that the data is usable, and that we can sort it and reduce the amount of enrollment, and I think that should be an effort. I'm not sure it's an MDIC effort, but I would vote for it, you're asking. But I think that's something that we need to address as an industry in diagnostics. And that along with that ties in with real-world evidence, and I think in a way it also ties in with digital health and wearables, where all that data would actually be usable in diagnostics, not just in the device.
Yeah, thank you. I think that, part of the question that I was asking today with the nest was how do we get access to that electronic health record data? I mean, IVDs have a little bit more complicated situations because we don't always know if our test has been modified as an LDT, if there's other issues in there. So, I think that's definitely
And if we don't start, we'll never.
issue. Exactly.
We'll never
Definitely an
get
issue.
there.
Yeah. And we'll have to think about how to resolve it. Jeff?
Yeah, so to deal with the issue that I would be concerned about, I think we would want to find a way to channel data from the labs back to the manufacturer or the sponsor so that we can get those data to FDA in some sort of supplement or 510K. Now that those data, will have their own challenges. And there are working groups that are dealing with this. I mean, we see Mike Waters at FDA, championing the semantic interoperability framework, and we have a seat at that table as well, and we want to figure out how best to explain to the labs that if they were to do this, could they prepare the data in such a way that one day we could circle it back through our process in the life cycle to get those claims on label, and then that should whittle down at this problem that we're seeing if it can be done in a predictable way.
Excellent. Jaime?
I think, similar comments, but I'll then say something a little bit different. So I think one of the other things for us, looking especially at the surrogate sample, the outline that was there, and I think one of the things that was up there was the sample stability. And I think, again, that's really a challenge for us and something that is not easily tackled. Certainly, we can consent people and get normal samples. Not so easy to consent to abnormal samples that span your measuring range for 21 parameters. So I think those types of things, looking at something along that line might be helpful for us as well.
Great. April?
There are a couple areas. One, in terms of new biomarkers, a tool in looking at how to bring those emerging biomarkers to a place, whether it's with reliance on, literature or concepts such as pre-market, post-market balance, what type of data might be useful in both settings as a way to help validate and move those new biomarkers. I agree with the comments about in silico samples. And approached, I think, a framework in that area, building upon the other activities of MDIC with patient computational modeling and virtual patient, would help with the industry. And then another would be just an assessment of, standards, the use of standards, and the role a standard can play with acceptance criteria in terms of clearly setting forth, expectations as to what is necessary to bring a product to market. And in part, I think about, existing technologies as maybe a place to start, recognizing that the more resources that are freed up from focusing in those well-established areas would then allow use of resources in these more innovative areas. And help promote getting those types of products to patients faster.
Terrific. Thanks. So Don, any more on your wish list? I know you gave us a few, but what's your wish list?
So I will talk about semantic interoperability again, because I think if the real-world evidence group is going to be successful, they're going to have to tackle that.
Mm-hmm.
Poor Tom sitting over there in the corner. He's like, oh, I can't believe he's saying it again. Tom, you know, it's like, okay. You know, and I are very singular sometimes in our approach. I think, I don't know, you know, I have, there are lots of things that haven't happened that I would have loved to happen. My industry is put on years ago, and I thought that would come up in these recent negotiations about letting, you know, doing like, I think it was the name that probably killed it, provisional approval or something. But it's an opportunity to actually have some data to say, yeah, this is probably going to be okay, and then you collect the data and you prove that it's okay, or you come off the market. So those concepts and I really like those as being drivers of innovation. So, but I think you're going to that digital health area and the whole research program.
Mm-hmm.
So I'm very much looking forward to seeing how that rolls out and plays out. Because a firm-based approach like that, really, I think you could. I think it would be a good way to manage resources internally and maintain a certain level of the entire industry that seems to be growing rapidly and is extremely innovative. So sometimes it's hard to keep up with that. So I'm very excited about seeing.
Great. Yeah, as are we. Any other thoughts from the panel before I throw out to see if anybody has any questions? Any other thoughts? No? Any questions from participants? Yes, I'm sorry.
So I think it's important to recognize the diagnostics group at MDIC as the youngest group, and that anyone who's in the room who doesn't currently participate, get involved, you're missing out on an opportunity to really make a difference. And so we need more people at the table. There's a lot of us up here have members on every single working group. It'll be great to hear more voices, get more input, get more data. I think that will make us stronger, make us grow. So a little bit of a cheerleading thing. Bill's nodding. Yep. And so if you don't belong to MDIC, join. If you do, if you are a member, then participate. Because you get the most out of it, and you actually get to work with a lot of great people and share a lot of great ideas.
Yeah, and I will actually second that. Roche was a latecomer to this group. We joined less than a year ago, and we already are embedded in a number of working groups and enjoying it, as well as finding it incredibly useful. So I told somebody today it's the best money we spend, but okay. Anyway, that's my pitch. So what I'd like to ask of you is, are there any questions that you have? Again, we have a shy audience. Yes, please.
I don't want to monopolize the conversation.
No, please do. Oh, perfect. Thank you, Carol.
We don't usually give the Zoom a mic.
Well, she was going to give him one that didn't work.
So first of all, thank you very much to the panel. Really appreciate it. And this question really is more open to the entire audience and everybody here, because I really would like to get a perspective. With respect to pediatrics, there are very few clinical diagnostics that have been initially evaluated prior to release to the wild or prior to release out to all hospitals. And yet across the country, across the globe, all of those clinical diagnostics that have been approved by the FDA are utilized for children. And for the, if you really think about clinical diagnostics, it truly is the backbone of medical practice in so many ways. Major medical management decisions are made based off of that information. There's a population that obviously hasn't been studied as well, but we've left it to the pediatric clinical community to evaluate those systems and understand how they will actually work for the vast array of physiologies and development in pediatrics. With respect to some of the concepts that have been discussed today, including the surrogate opportunities, do you see a way where we can begin to more effectively evaluate clinical diagnostics for children prior to their release, or in some balanced method with a pre-market, post-market balance that allows for more evidence in pediatrics and clarity about how these devices work for pediatrics?
Okay, I'm going to throw that first to the panel. Does anyone on the panel want to speak to that? Yeah?
So,
yeah,
I can say, I mean, I think it's a very interesting question. I think it's something we struggle with, definitely. When you're looking at residual samples from pediatric patients, it's very difficult when it's a fresh sample for the site to actually give that up for a study. And if you're talking about a neonate, even more rare that they're going to give you a specimen to be able to take over and run through your, device. That really, for them, is probably not going to provide any benefit to that individual subject, right? And then to try to run a study where you would be consenting to get those samples, even more difficult, right? If you think of a neonate and you don't need to draw blood from them, is the IRB going to prove that? You're going to draw more blood from them in order to do a study. And so I think it's a challenge that we definitely run into, and we were just kind of kicking around, how do we use going forward? Is it real-world evidence that we use, once the device is on the market, to start looking at that? So I think it definitely is a struggle for us. How do we do that? Or do we get approved, like you're saying, and then do it as post-market, looking at it that way?
Don, have you seen any good experiences within FDA looking at pediatric diagnostics?
So I guess
and you can reframe the way I asked that question.
So I think in our chemistry, I think you have to be a little bit creative when it comes to pediatrics, especially neonates.
Yeah.
So you've got to look at those in creative ways. I think in the diabetes area, and everything populations, but I do. It is difficult. Maybe a form of real-world evidence is going to be the way that I don't have any great input.
Okay, but thank you. It sounds like there are some that are doing it, and I know it's a challenge for all of us. We actually are out of time. I just got the 0 minutes left. So thank you very, very much to the wonderful panel and to all of you for listening so intently and for your questions. Thanks so much.